Somewhere in a genetics laboratory in Haifa, Israel in 2009, a research team led by a molecular geneticist named Doron Behar sat down with a dataset that was, at that point, the most comprehensive genetic survey of Jewish populations ever assembled. They had collected DNA samples from individuals across 14 Jewish communities: Ashkenazi Jews from Eastern Europe, Sephardi Jews from Turkey and Bulgaria, Mizrahi Jews from Iraq and Iran and Yemen, North African Jews from Morocco and Libya, and communities from the furthest geographic fringes of the diaspora, including communities from Ethiopia and India. And they had compared all of it at nearly 500,000 genomic locations simultaneously against the DNA of 69 non-Jewish populations drawn from across the planet. What they were looking for was the genetic architecture of a people who had, by every historical account, spent the better part of 2,000 years scattered across every continent, living as minorities inside dozens of different civilizations, speaking dozens of different languages, and absorbing, to varying degrees, the genetic contribution of every population they lived among. What they expected to find was the genomic evidence of that dispersal. Distinct regional clusters, significant genetic distance between communities, the gradual dilution of any shared ancestry across 2 millennia of diaspora. What they actually found, when the data resolved, was something that stopped the scientific community cold, not because it was impossible, not because it required any revision of the underlying biology, but because it demonstrated, with a precision that no historical record alone could have achieved, that a group of people dispersed across more than 2,000 years and 6 continents had, against every demographic expectation, maintained a genetic coherence that was, in most cases, stronger across the distance between them than the coherence between each of those communities and the non-Jewish populations they had lived beside for generations. A Polish Jew and a Moroccan Jew, separated by geography, language, religious tradition, and centuries of completely independent community history, were, by the measurement of these 500,000 genomic markers, more closely related to each other than either was to the Polish or Moroccan neighbors their communities had lived among for as long as anyone could document. The diaspora, as a genetic event, had not done what the diaspora, as a historical event, should have done. The people had scattered. Their DNA, to a remarkable and unexpected degree, had not. This is the real story of what happens when you apply the full toolkit of modern population genetics to the oldest and most continuous ethno-religious community in documented human history. Not a viral claim about a fabricated discovery. The actual findings from a decade and a half of peer-reviewed research, published in Nature and Nature Communications and the American Journal of Human Genetics, by teams of researchers at Rambam Medical Center and Columbia University and Hebrew University of Jerusalem, who were not looking for a dramatic conclusion and found one anyway. To understand why those findings are as surprising as they are, you first need to understand what the researchers expected to find based on what historical dispersal at this scale normally does to a population's genetics. The mechanism is straightforward. When a population fragments and its pieces migrate to geographically separated regions, each fragment is subject to the genetic influence of whatever populations it lives among, through intermarriage, through conversion, through the ordinary demographic processes that blur the boundaries between communities over time. The longer the separation and the more contact with local populations, the more the original shared genetic signature gets diluted. Over 2,000 years, across 6 continents, the expectation was that this dilution would have proceeded far enough to make the original signal difficult or impossible to distinguish cleanly from the surrounding genetic noise. The Behar study found that it hadn't. Specifically, it found that most Jewish diaspora communities, the Ashkenazi, Sephardi, Mizrahi, and North African populations, clustered together in genetic space in ways that showed their shared Levantine origin more clearly than their regional differences. Multidimensional scaling analysis, which maps the relative genetic distances between populations in visual space, placed Jewish communities from across Europe, the Middle East, and North Africa in close proximity to each other and to modern populations from the Levant and Cyprus, significantly closer than any of them were to the non-Jewish populations of the countries they had inhabited for centuries or millennia. The conclusion the study's authors drew was specific and carefully worded. The results trace the origins of most Jewish diaspora communities to the Levant and reveal that the shared genetic ancestry of these communities is identifiable across vast geographic separation. What this means in concrete terms is that Jewish endogamy, the practice of marrying within the community, maintained across 2,000 years by religious law, social structure, and the enforced separation of Jewish communities from their surrounding societies by a combination of Jewish tradition and external legal discrimination, left a detectable and consistent genetic signature that modern sequencing technology can read directly. The historical practice of maintaining community boundaries, enforced from both inside and outside, shows up in the genome as a coherence that preserved the original Levantine ancestry while limiting, rather than eliminating, the genetic contribution of the surrounding populations. This is the first finding, the one that makes the distance between an Ashkenazi Jew from Warsaw and a Sephardi Jew from Morocco genetically shorter, in a measurable and reproducible sense, than the distance between either of them and their respective non-Jewish neighbors. It is not a finding that either confirms or contradicts any particular religious or political position about Jewish identity. It is a finding about population genetics, about what 2,000 years of demographic practice produces in a genome when that practice is consistent enough and widespread enough to be statistically visible at the level of 500,000 genomic markers. The second finding is the one that researchers who cover population genetics describe as the most startling single number to emerge from any Jewish genetic study. It concerns specifically the Ashkenazi Jewish population, the community that traces its origins to medieval Central and Eastern Europe, from which roughly 2/3 of all Jews alive today descend, representing approximately 10 million people living primarily in Israel and the United States. The Ashkenazi founder effect, first characterized in detail by a 2014 study led by Shai Carmi at Columbia University and published in Nature Communications, produced a conclusion that has been replicated by multiple subsequent studies, including one using ancient DNA from a medieval Jewish cemetery in Erfurt, Germany, excavated in 2022. The conclusion is this: the entire Ashkenazi population of roughly 10 million individuals traces its ancestry to a founding group of approximately 350 people who lived somewhere in medieval Europe between 600 and 800 years ago. Not 350 families, not 350 ancestral lines, 350 individual human beings whose specific genetic contribution to subsequent generations was so dominant that when researchers compare the complete genomes of unrelated Ashkenazi Jews from different countries today, they find segments of identical DNA that are substantially longer and more numerous than equivalent segments found in non-Jewish European populations of comparable size. The length of these shared segments is a direct indicator of the recency of common ancestry. Longer identical blocks mean more recent shared ancestors, because each generation of reproduction breaks these inherited segments into shorter pieces. The blocks found in Ashkenazi genomes are long enough and shared widely enough that Itzik Pear of Columbia, one of the study's authors, summarized the finding to journalists with one of the clearest statements ever made about a finding in population genetics. Among Ashkenazi Jews, everyone is a 30th cousin. 350 people, a group that could fit inside a modern movie theater. The effective founding population of a community that would, over the following 6 to 8 centuries, expand to 10 million individuals and produce a disproportionate share of the scientists, writers, musicians, mathematicians, economists, and public figures who shaped the modern world. The historical context for this bottleneck is not mysterious. The 12th and 13th centuries were a period of intense, sustained, violent persecution of Jewish communities in Western Europe. Massacres during the Crusades, expulsions from England in 1290 and France in 1306, the beginning of a series of legal and social restrictions that would eventually confine Jewish communities to specific geographic zones called ghettos. The surviving community that fled eastward into Central and Eastern Europe, where conditions were initially somewhat more permissive, was numerically small and genetically cohesive. From that small group, sustained by endogamy and geographic concentration, came the entire Ashkenazi population. The Erfurt Cemetery study, published in 2022, added a specific and precise piece of evidence to this picture. Researchers extracted and analyzed the genomes of 33 individuals buried in a 14th century Jewish cemetery in Erfurt, Germany, a community killed during the Black Death pogrom of 1349, when Jewish communities across Central Europe were massacred in the belief that they had poisoned wells to cause the plague. The genomes of these medieval Erfurt Jews were genetically similar to modern Ashkenazi Jews, confirming that the founder event and the acquisition of the characteristic Ashkenazi genetic signature had already occurred before the 14th century. 8 of the 33 individuals carried pathogenic genetic variants known to affect modern Ashkenazi Jews today. Variants like the BRCA1 mutation associated with elevated risk of breast and ovarian cancer, present in the Ashkenazi population at a significantly higher rate than in the general population. A direct consequence of the founder effect, amplifying whatever variants happened to be present in those 350 original individuals. This last point is the one that connects the findings of Jewish genetics most directly to medicine. The Ashkenazi bottleneck did not only preserve ancestral identity, it also amplified certain disease-associated genetic variants simply by chance, because the founding population happened to carry them, and because subsequent endogamy and population growth spread those variants through a community that was expanding rather than mixing them out through contact with a broader gene pool. Tay-Sachs disease, Canavan disease, Gaucher disease, familial dysautonomia. a set of conditions that are significantly more prevalent in the Ashkenazi population than in the general population, not because of any particular property of Jewish ancestry, but because of the straightforward genetics of a small founding group followed by rapid expansion within a largely closed community. The same process that made everyone a 30th cousin also concentrated the variants that were present in those 350 people. And then there is the finding that complicates the entire picture, because it forces a question that population genetics is capable of posing, but not capable of answering. What does it mean to be genetically Jewish? The Behar study included, alongside the Ashkenazi, Sephardi, Mizrahi, and North African communities, 3 Jewish populations from the furthest geographic fringes of the historical diaspora. The Beta Israel of Ethiopia, Jews who have practiced a form of Judaism for centuries and who maintain a deep and sincere religious identity, including observance of the Sabbath, dietary laws, and Jewish festivals derived from the Hebrew Bible. The Bene Israel of Western India, another ancient community with a documented Jewish identity stretching back many centuries, and the Kochan Jews of southern India. When the researchers ran these populations through the same comparison they had applied to every other community, the results separated them sharply from the pattern they had found everywhere else. The Beta Israel, the Bene Israel, and the Kochan Jews did not cluster with the other Jewish communities. They clustered with their neighbors, with non-Jewish Ethiopians in the case of the Beta Israel, with non-Jewish Indians in the case of the Bene Israel and Kochan Jews. The shared Levantine genetic signature that was visible and consistent across the Ashkenazi, Sephardi, Mizrahi, and North African communities, the genetic thread connecting a Warsaw Jew to a Casablanca Jew, was absent or minimal in these 3 populations. Genetically, they were Ethiopian and Indian first, and connected to the Middle Eastern origin of other Jewish communities only faintly or not at all. The most comprehensive modern analysis of Beta Israel genetics, using the Global 25 database of ancient and modern population genomes, placed the average Beta Israel individual at a genetic distance from the Amhara and Tigrayan populations of the Ethiopian highlands that is smaller than the distance between Amhara individuals themselves. In other words, closer to their Ethiopian neighbors than those neighbors are to each other. The nearest Jewish population on Earth, the Yemenite Jews, sits roughly 27 times further from the Beta Israel, genetically, than the Amhara do. A study published in 1999 by researchers at the International Institute of Anthropology in Paris explicitly concluded that the Beta Israel descended from ancient inhabitants of Ethiopia, who converted to Judaism, rather than from Levantine immigrants. The conversion hypothesis, that these communities adopted Jewish religious practice without descending from the populations that gave rise to the broader diaspora, is the conclusion strongly supported by the autosomal genetic data. This finding does not settle the question of who the Beta Israel are or what their relationship is to the broader Jewish world. The State of Israel recognized the Beta Israel as Jews under the law of return and brought them to Israel in Operation Moses in 1984 and Operation Solomon in 1991. Their Jewish identity is legally, religiously, and historically recognized in ways that have nothing to do with genome-wide S&P data. But what the genetics shows, clearly and reproducibly, is that Jewish identity, the category the research was built to investigate, does not map neatly onto a single genetic signature. It is a category that includes communities whose shared ancestry is traceable in the genome, communities whose shared practice is traceable in the historical and religious record, and these are not the same set of communities. And the category cannot be reduced to either one alone without losing something essential. This is where the research lands in territory that makes everyone uncomfortable in a specific and instructive way. The finding that most Jewish diaspora communities share a traceable Levantine genetic origin, maintained across 2,000 years of dispersal, is a finding that both confirms one aspect of Jewish historical experience, the idea of a people with a common origin, and simultaneously defies the categories that modern genetics has elsewhere dismantled. The earlier script in this series showed that the concept of race has no meaningful basis in population genetics, that the variation within any so-called racial group is greater than the variation between groups, that the categories used to divide humanity have no consistent genetic foundation. Jewish identity does not fit cleanly into that framework in either direction. It is not a racial category. The genome-wide data shows non-Jewish populations across the Middle East sharing more overall genetic similarity with Jewish populations than many Jewish populations share with each other across the Atlantic. But it is not purely a cultural category either, because the genetic signal is measurable, consistent, and reproducible across 14 communities on 3 continents. What the data actually describes is a category that does not correspond to any pre-existing classification scheme, not race, not ethnicity in the conventional sense. Not purely religion, something that has no good single-word name in English, a combination of shared ancestry, shared practice, shared historical experience, and shared endogamy that has produced a measurable genetic pattern without producing anything like the clear genetic boundary that racial categories have always implied. Harry Oster, a geneticist at New York University and one of the field's leading figures in Jewish population genetics, described it in a 2010 review as a hereditary persistence, a maintenance of a specific ancestral signature through a specific social and demographic practice, sustained across a span of time that makes it unique in the documented history of human population movement. What the comparison to populations worldwide ultimately shows, then, is not what the title of a viral video promises. It does not show that Jewish people are genetically superior, or genetically separate, or genetically anything that maps onto the categories that bad faith actors have historically tried to assign to genetic data about Jewish populations. It shows that a specific set of historical practices, maintained endogamy, community cohesion, religious law governing marriage, and centuries of forced separation from surrounding societies, produced a genetic signature that is measurable, consistent, and in most cases stronger across the distance of the diaspora than across the boundaries between Jewish and non-Jewish communities living in the same geography. And it shows that even that measurable signature has limits, that the communities at the edges of the diaspora, the Ethiopian Jews who observe the Sabbath in Amharic, the Indian Jews who recite Hebrew prayers with Marathi accents, demonstrate that Jewish identity extended, in some times and some places, through religious conversion and cultural adoption, rather than genetic descent, producing communities whose commitment to the practice is indistinguishable from communities whose ancestry runs back to the ancient Levant, while their genomes tell a completely different origin story. The result, when you lay it all out, is a picture of a single human group that managed to do something almost without parallel in the population genetics of the modern world, maintain a measurable genetic connection across 2,000 years, 6 continents, dozens of languages, and the full sweep of Western and Middle Eastern history, through nothing more technologically sophisticated than the consistent social practice of marrying within the community. No mechanism, no intervention, just an accumulated demographic choice, repeated generation after generation across 2 millennia, that left a signal in the genome precise enough for modern sequencing technology to read and compare against 69 other populations worldwide. That signal is real. What it means about identity, about history, about what connects a family in Warsaw to a family in Casablanca to a family in Baghdad and back through 2,000 years to the same set of hills in the Eastern Mediterranean, is a question that genome-wide SNP data can illuminate but cannot answer, because the data can tell you where the ancestry came from. It cannot tell you what that ancestry means to the people who carry it, subscribe, because this is the kind of science that puts numbers on questions that humanity has never been able to stop asking.